Dear students, welcome to my channel. The topic is Northern Blotting. Before going to the topic, please subscribe my channel. Press the bell icon for notifications. Like, share among your friends and comment. The Northern Blot technique was developed in 1977 by James Alvin, David Kemp and George Stank at Stanford University. The technique gets its name due to the similarity of the process with the Southern blotting. The primary difference between these two techniques is that Northern blotting concerns only about RNA. Northern blotting is a technique used for the study of gene expression. It is done by detection of particular RNA or isolated mRNA. mRNA is generally represented as 5% of the overall RNA sequence. This method reveals identity, number, activity and size of the particular gene. Blotting technique can also be used for the growth of a tissue or organism. In different stages of differentiation and morphogenesis, the abundance of RNA changes and this can be identified using this technique. It also aids in the identification of abnormal disease or infected conditions at the molecular level. When we examine the principle of Northern blotting, start with the electrophoresis to separate RNA sample by size. Electrophoresis separate the RNA molecule based on the charge of the nucleic acids. The charge in the nucleic acid is proportional to the size of the nucleic acid sequence. Thus, the electrophoresis membrane separate the nucleic acid sequence according to their size of RNA sequence. In the case where on target sequence is in mRNA, sample can be isolated through oligocellulose chromatographic technique as mRNA are characterized by the poly A tail. Since gel molecules are fragile in nature, the separated sequence are transferred to the nylon membrane. The selection of nylon membrane is contributed to the factor that nucleic acids are negatively charged in nature. Once the RNA molecules are transferred, it is immobilized by covalent linkage. The probe is then added. The probe can be complementary as single-stranded DNA sequence. Formamide is generally used as a blotting buffer as it reduces the annealing temperature. The principle of the method is the separation of RNA according to size by agarose cell electrophoresis and afterwards transferring the separated RNA on a nylon membrane. Consequently, the detection of the desired RNA is done by using the radio-labeled probe with all or a part of the base sequence of the targeted RNA hybridizing with the immobilized and separated RNA. These are the materials and equipment required for Northern blotting. Next, we can see the procedure of Northern blotting. The first step is RNA extraction and separation. Second, transfer of the separated mRNA. Then, fixing the separated RNA in the membrane. Radio labeling with the probe. Hybridization and analysis. The first step is RNA extraction and separation. Firstly, prepare an agarose gel and pour it into a casting tray. Then let the gel solidify. After that, run the running buffer for equilibration for about 30 minutes. Simultaneously, mix the sample with RNA loading buffer. Then add RNA marker to the mixture above and incubate. After that, mix RNA sample mixture in the equilibrated gel. At last, run the gel at 125 volt for approximately 3 hours. Next step is transfer of the separated RNA. Cut a nylon membrane and 3 mm Whatman paper in the same size as denaturation gel. Afterwards, place the gel above a sponge for support in a vacuum container containing SSC, that is called transfer buffer. Then position the agarose gel in such a way that the separated RNA faces above. After that, place the cut and wet nylon membrane on top of the gel. Similarly, place the Whatman paper above the membrane. Then vacuum pressure is provided in the container. The estimated time for the transfer is 90 minutes. Next step is fixing the separated RNA in the membrane. After the transfer completes, cross-link with UV to fix the RNA in the membrane first. Then repeat the cross-linking process. Radio labeling the probe. Firstly, prepare probe similar to the target RNA with NTPs. Then radio-label the probe. Finally, introduce the probe to the membrane. Hybridization and analysis. Hybridize the probe by placing the membrane in hybridizing solution. Then wash the membrane after hybridization. Finally, analyze the membrane under X-ray and use software for quantification. This figure shows the summary of Northern blotting. Sample RNA is extracted, subjected to electrophoresis, then RNA separated by the size. Northern blotting, that means transfer of RNA to membrane, then RNA fixed to membrane with UV or heat. Labeled probes are added, then so membrane hybridized with labeled probes will get. Then visualization of labeled RNA on X-ray film. Next is advantages and disadvantages of Northern blotting. The advantages of Northern blotting help in the detection of small changes in RNA. It has high specificity. It is also a quantitative method. The disadvantages, as it has less sensitivity. Also, maintaining an RNA's free medium is challenging. Likewise, analysis of a large number of genes becomes tedious. Dear students, thank you for watching. See you next class with another topic. Don't forget to leave a like and please subscribe my channel.